DNA is often presented as fate. We inherit eye color, height, and disease risks, and from this, it is easy to imagine our biological outline is predetermined at birth. Cultural shorthand reinforces this idea by speaking of the gene for anxiety or obesity. This framing suggests the body is simply carrying out a fixed script written long before we arrived. This view treats the body as a machine executing static commands, overlooking the biological processes that determine how those instructions are actually used. Nearly every single cell in the complex architecture of the human body contains an exact, identical set of genetic instructions. Despite sharing this genome, these cells maintain radically different identities. As this diagram shows, a neuron and a skin cell possess the same code, yet they produce different proteins and perform different functions. This occurs on a human scale as well. Identical twins begin life with matched DNA. Yet decades later, these same twins often diverge in metabolic health, disease risk, and cognitive function. Their genes remain matched, but their biological outcomes do not. The DNA sequence stays static, while the way it is read changes. This layer of biological management fills the gap between the code we inherit and the life the body actually experiences. Epigenetics refers to the regulatory systems that influence gene expression without altering the underlying DNA sequence. It determines which portions of the genetic library are amplified or silenced. Researcher David Sinclair compares the genome to hardware and the epigenome to the software that determines how that architecture operates from one moment to the next. Sinclair also uses the image of a piano. The genome provides the keys, while the epigenome acts as the pianist, deciding how loudly, softly, or rhythmically those keys are played. Biological identity emerges through this continuous interpretation of code in response to external conditions. Cells establish their identities by selectively activating specific pathways while suppressing others, eventually stabilizing into skin, muscle, or nerve tissue. One primary tool is DNA methylation. This involves attaching small chemical methyl groups to the DNA molecule. These markers function like a dimmer switch. Regions with high methylation are generally less active, signaling the cell to ignore those specific instructions. Physical packaging also plays a role. DNA wraps around proteins called histones, allowing several feet of genetic material to fit inside a microscopic nucleus. When this structure winds tightly, genes become inaccessible. When it relaxes, genes become accessible for activation. Genes exist within a responsive architecture that continuously changes its shape to manage which information is executed. Environmental signals reach deep into this molecular machinery, dissolving the perceived boundary between the outside world and internal biology. External factors like stress, our daily nutrition, and social experience trigger chemical pathways that reach the methylation and histone systems to influence gene expression. This is visible in early development. Research involving maternal care in rats found that pups receiving high levels of grooming developed epigenetic changes that regulated their cortisol receptors, leaving them more resilient to stress as adults. Nutrition acts as a similar signal. In agouti mouse studies, specific nutrients in the mother's diet shifted the coat color and obesity risk of her offspring by altering methylation patterns, while the DNA sequence remained unchanged. The gut microbiome also participates in this conversation. Bacteria convert dietary fibers into signaling compounds that inhibit the enzymes responsible for DNA packaging, influencing inflammation and immunity. Nurture regulates nature through these chemical pathways. Over a lifespan, the body's ability to maintain these precise expression patterns begins to fluctuate. The information theory of aging suggests that the decline we associate with getting older is driven by the accumulation of epigenetic noise. As this data visualization shows, cells gradually lose the precision of their dimmer switches, regulatory drift occurs, and cellular identity becomes less stable. Aging reflects an issue of instruction management. Cells slowly lose the memory of how to remain themselves. Genetic vulnerabilities and inherited risks remain a foundational part of our biology. However, a genetic risk represents a biological possibility rather than a guaranteed outcome. Sleep, movement, social connection, and nutrition act as continuous signals to the regulatory software throughout adulthood. The body determines how the code is expressed in response to the environment we inhabit. Biological inheritance functions as an ongoing dialogue with the environment, leaving the organism adaptive and responsive to the lives we lead.
Thank you for watching. If something in this resonated with you, then you're already closer than you think. If you're ready to go deeper, there's a link below to my book, The Survival Self: How Childhood Adaptation Becomes Adult Identity, and How to Finally Live Beyond It. This isn't theory, it's a framework you can actually use. Tap the link below and step into it.